Today, we're going to unravel a pretty amazing scientific detective story. It comes from research out of the Washington University School of Medicine, and it's a story about a truly devastating disease, a clue hidden deep inside our DNA, and, well, a potential breakthrough that could change absolutely everything.
So, let me just start with this question. What if a fatal brain disease, one that has stumped scientists for decades, could be stopped? Not by some super complex procedure, but by just flipping a single switch inside our cells. Sounds like science fiction, right? Well, maybe not. To really get how huge this switch idea is, we first have to understand the puzzle that researchers have been staring at for years. And it is a very complex puzzle. Okay, so Huntington's is a really cruel disease. It's inherited, it's fatal, and there's no cure. It's caused by a single faulty gene that just relentlessly breaks down nerve cells in the brain, affecting how a person walks, thinks, and even feels.
Now here's the crazy part. Scientists have known who the culprit is for a long time. It's this toxic mutant protein called Huntington, or HTT for short. But, in any investigation, knowing who the bad guy is isn't enough. The real mystery has always been how. How exactly does it kill brain cells? What's the murder weapon? The first big clue came when they noticed something really disturbing happening. It turns out this mutant HTT protein acts like a saboteur inside our cells. It forces critical genes to just go silent. We're talking about genes like BDNF, which is basically fertilizer for your brain cells, keeping them healthy, and other genes that are essential for everything to work right. They were just being switched off. The scientific term for this is transcriptional dysregulation. And that clue, that gene silencing, it led researchers to look in a new direction. Maybe the problem wasn't with the genes themselves, the hardware, but with the system that was controlling them, something like a genetic control panel. And that brings us to this fascinating field called epigenetics. The easiest way to think about it is this. If your DNA is the computer hardware, epigenetics is the software. It's this whole other layer of instructions that tells your genes when to power on and when to shut down. It's the control panel running the entire show without ever changing the DNA code itself. So one of the main tools this epigenetic software uses is called DNA methylation. Just picture it like a tiny chemical sticky note that gets slapped onto a gene. And when that note is there, it's a clear signal to the gene, hey, you need to be quiet. It acts like a dimmer switch, turning the gene's activity way down or sometimes turning it off completely.
And the way it works is actually pretty straightforward. There's this little worker enzyme called a DNA methyltransferase. Let's call it a DNMT. It zips over to a gene, it attaches that little methyl tag, that sticky note, and boom. Just like that, the gene is silenced.
And right there, that's the big question, isn't it? Could this be the murder weapon they were looking for? Was it possible that the mutant Huntington protein was hijacking these little DNMT enzymes, basically forcing them to go around sticking off notes on all the wrong genes, leading to the death of the brain cell? Okay, so to test that theory, the researchers set up this absolutely brilliant experiment. It was a massive drug screen designed to see if they could find something, anything, that could block the toxic protein from killing the cells. And when I say massive, I mean it. They didn't just test one or two things. They systematically went through 84 different drugs. Each one was designed to mess with a different part of that epigenetic control panel, all to see if one of them could save the neurons. And the results for one of those drugs, well, they were nothing short of dramatic. Here's what they saw. You take a neuron with the mutant HTT protein, and, as expected, it dies. But, and this is the amazing part, when they added a specific drug called decitabine to the mix, the neuron survived. The drug gave it almost complete protection from that killer protein.
Now here's the thing. Decitabine is a known blocker of those DNMT enzymes we were just talking about. So that was a huge lead. But, good detectives, they need more than just a lead. They need proof. They had to be sure this was the real mechanism.
So they came at it from two different angles, to be sure. First, the drug approach, the pharmacological method, where they used decitabine to physically block the enzymes from working. And then a totally different genetic method. They used a cool technique called RNA interference to basically tell the cell's own factory to just stop making so many of those DNMT enzymes. And the result? It was exactly the same. Whether they blocked the enzymes with the drug or just got rid of them genetically, the neurons were protected. This was the smoking gun. It proved that the mutant protein needs those enzymes to do its damage. This whole DNA methylation thing wasn't just involved, it was absolutely required for the brain cells to die.
So, okay, if blocking this silencing mechanism saves the cell, does it fix the original problem, right? Does it actually turn those essential genes back on? Well, let's look at that crucial brain fertilizer gene, BDNF. You can see it clear as day on this chart. The first bar, that's a healthy neuron, normal gene activity. Now look at the second bar, that's the Huntington's neuron. Activity just plummets. But then, look at bar number 3. When they add the drug, bam! The expression level shoots right back up to normal. They were literally turning the lights back on.
And how did it work? It fixed the exact problem they suspected from the start. The drug went in and basically peeled off all those wrong sticky notes that had been slapped onto the gene's control switch. And once those were gone, the gene could get back to doing its job. And this is so important. These amazing results weren't just happening in a petri dish. The researchers did the same thing in the brains of mice that were engineered to have Huntington's. And you see the exact same pattern. Low gene levels in the sick mouse and restored levels in the sick mouse that got the treatment. It actually worked in a living brain. That is a huge deal.
So where does all of this leave us? What does it all mean for the future? Could this discovery have just blown open a brand new door for treating this disease? I mean, let's just recap the whole detective story. The crime? This mutant protein is killing brain cells. The murder weapon they were searching for? Turns out it was gene silencing, done using DNA methylation. And the solution? Block the weapon with these DMMT inhibitor drugs. Case closed.
The actual scientists who did this work, they put it perfectly in their paper. They said that messing with this DNA methylation pathway could be an attractive new therapeutic strategy for fighting Huntington's. It's an understatement, but a powerful one. And that leaves us with one last really big thought. For decades, we've been trying to figure out how to fix problems in our genetic hardware. But what if the real future, the real frontier, is learning how to edit and debug our genetic software? This incredible discovery in Huntington's might just be the very beginning of that new era.